am Tirtha Mukherjee, and today I am excited to share my project on studying the bioactivity of aspirin, menthol, and vanillin through computation. This project isn't just about these compounds, it's a journey into understanding how to predict and study bioactivity of compounds through computational methods. In my fourth semester of MSc Chemistry, I have delved into this topic, and today I will share some glimpse of it with you. Before I begin, I want to express my gratitude to our respected principal sir and our external expert for being here today. I also extend my gratitude to our course coordinators and dedicated instructors for their unwavering support and guidance throughout this project. So without further ado, let's get into it. At first, let's quickly go over the contents of today's presentation: introduction, objectives, methodologies, main findings, conclusion, and references. Let's begin our introduction by exploring the concept of bioactivity. In simple terms, bioactivity refers to how a substance like a drug or a chemical compound affect living tissues. It's all about understanding the impacts these substances have on our bodies. In pharmacology, the focus is on studying the beneficial or adverse effect of drugs on biological systems. And these are the references. Moving on, these are the bioactive compounds I have chosen: aspirin, menthol, and vanillin. So let's begin with aspirin. This is the structure of aspirin. So aspirin is a non-steroidal anti-inflammatory drug synthesized from salicylic acid. It works by inhibiting the enzyme cyclooxygenase, which is responsible for the synthesis of prostaglandins. Prostaglandins are lipid compounds that play a role in pain, inflammation, and fever. So by inhibiting COX, aspirin reduces the production of prostaglandins, thereby alleviating pain, reducing inflammation, and lowering fever. And these are the references I have referred. Moving on, aspirin is widely used for several purposes. Firstly, pain relief. Aspirin is most commonly used to relieve mild to moderate pain. It is also effective in reducing fever. It alleviates inflammation. It is used as a preventive measure for cardiovascular diseases. And if aspirin is taken in low dosage, it also helps in blood coagulation. These are the references. Moving on to our next compound, that is menthol. This is the structure of menthol and its scientific name. So menthol is a naturally occurring compound found in peppermint and other mint plants, known for its cooling, minty characteristics. It is used in various products for its sensory effects, analgesic properties, and ability to affect physiological processes such as muscle contractility and neurotransmitter receptor activity. So these are the images of some peppermint or mint plants: peppermint, spearmint, wild mints, etc. And these are the references. Now, the next is uses of menthol. Menthol is commonly utilized to alleviate cough and congestion, providing relief for nasal passage congestion. It also had analgesic properties. It alleviates inflammation, aiding, and it serves as an antioxidant, protecting cells from oxidative stress. The references. Moving on, our next compound is vanillin. Now, this is the structure of vanillin and the isotope. The vanillopores contain a vital ingredient called vanillin. This is the picture of vanillopores. Now, vanillopores contains a vital ingredient called vanillin, and that acts as a component of fragrance. It is an extensively researched molecule, also known for its sweet, soft, and slightly spicy qualities. For this reason, it is used in numerous fields such as food industry, perfume industry, and medical establishment. These are the references. Moving on, the uses of vanillin. Vanillin is widely used in many diverse fields. Firstly, as a valuable renewable chemical, vanillin is often used as an ingredient in bio-based polymers due to its aromatic properties. Secondly, it serves as a flavored compounds. These are some of the flavored compounds where vanillin are used: chocolates, ice cream, creams, etc. It also acts as an antioxidant, improving the quality of food products, rich in polyunsaturated fatty acids. Finally, vanillin possesses neuroprotective effects and could have potential therapeutic uses in pathophysiological conditions. These are some of the neuropathophysiological conditions such as Alzheimer's disease, Parkinson's disease, etc. And the references. Moving on to the objective of our project. So for this project, I utilized ChemTro and Pass Online to analyze the bioactivity of the chosen compounds and conducted a thorough literature survey and explained them in this project. I have found these following bioactivities for my compounds. Now, let us discuss the methodologies I have used to analyze the bioactivity of a chosen compound. In step 1, I have used ChemTro window for drawing the compounds. In step 2, I have logged in to the Pass Online website and clicked on Go for Prediction. In step 3, I have to click on Predict New Compound and then Marvin chess. Then after clicking Marvin chess, this Marvin chess window will appear. In that, we have to click on this open folder and we have to select the .mol file of the compound chosen and the structure of the compound will appear here. In step 6, we have to click on Predict and we will finally get to see the predicted bioactivities of our chosen compound. Last but not the least, the final step is to do the literature survey of the bioactivities and gather information about them. Now, moving on to the main findings of our analysis. Firstly, bioactivities chosen for aspirin from Pass Online website. First is fibrinolysis. Fibrinolysis is the process where fibrin in blood clots is broken down, helping control blood coagulation and maintain vascular health. Now first let us understand what is a blood clot. Blood clots, also known as thrombi, are clumps of blood that have changed from liquid to a gel. That transformation, also called coagulation, this transformation is known as coagulation. This happens to stop the damaged blood vessels from leaking blood either at the skin or inside the body. So, blood clotting or coagulation is an important process that prevents excessive bleeding when a blood vessel is injured. Platelets are a type of blood cell and proteins in your plasma, the liquid part of the blood, work together to form a clot made from a substance called fibrin. Now, if you can see the fifth number, the yellow thread-like structure is called the fibrin, and this clot develops into a scab that protects the wound as it heals. After bleeding has stopped and healing has occurred, the body should break down and remove the clots. However, sometimes blood clots form too easily or don't dissolve properly and travel through the body, limiting or blocking the blood flow. This is where fibrinolysis comes into play. It involves a series of reactions that breaks down fibrin and dissolve blood clots, preventing thrombotic diseases. Now, these are some thrombotic diseases like stroke, heart attack, deep vein thrombosis, especially seen in legs of human beings, and the references. Moving on to our next bioactivity, antipyretic. Antipyretics are drugs that help reduce fever. They work by stopping the enzymes that make prostaglandins. Now, what are prostaglandins? Prostaglandins are chemicals in the body that controls things like the immune system and the body temperature. So, by stopping the enzymes that helps in production of prostaglandins, antipyretics can lower fever and make you feel better. And these are the references. Moving on to antiseboric. Antiseboric refers to a substance or treatment that prevents or relieves the symptoms of seborrhea or seborrheic dermatitis. Now, these conditions involve excessive secretion of sebum, an oily substance produced by the skin's sebaceous gland. As you can see in this picture, this is the sebaceous gland, and this is where the sebaceous gland is present. And the number 4 mark here is the sebum. Excessive secretion of sebum produces disease. This is how seborrhea or seborrheic dermatitis looks like. It is a chronic skin disease that causes flaky, scaly, greasy, and occasionally itchy and inflamed skin. Seborrheic refers to the sebaceous glands, while derms mean skin. And these are the references. Moving on to antiseptic. Antiseptic are chemicals that prevents or fight infections by stopping the growth or killing microorganisms on skins or mucous membranes. They are used in hospitals and surgeries to reduce bacteria, fungi, and viruses on surfaces and skins. The references. Moving on. Next is anti-inflammatory. Anti-inflammatory agents are substances that reduce inflammation. Now, inflammation is a body's immune system's response to an irritant, such as germ, foreign object, or damaged cells. It is a normal part of healing process when the body is injured or infected. Anti-inflammatory agents helps reduce this response, easing symptoms and aiding in the healing process. And the references. Next comes the bioactivities chosen for menthol from Pass Online website. First is vasoprotectors. Vasoprotectors are agents that help protect the vascular system from damage and maintain its proper functions. They do this by shielding blood vessels from harm through various mechanisms, including influencing the lipid metabolism. Now, lipid metabolism is a process of synthesizing, breaking down, and storing fat in cells for energy storage. These are the references for this slide. Moving on. Next is anti-eczematic. An anti-eczematic is a medication or treatment used to alleviate the symptoms of eczema, also known as atopic dermatitis. Now, this is what eczema looks like. Atopic comes from the word atopy, which describes a tendency to respond immunologically to allergens. And dermatitis comes from the Greek word derma, which means skin, and itis, which means inflammation. So eczema is a common chronic skin disease characterized by dry, itchy, and inflamed skin. It causes redness, irritation, scaliness, and can affect various parts of the body, including face, arms, legs, hands, and feet. And I've referred to this for the slide. Next is sugar phosphatase inhibitor. A sugar phosphatase inhibitor is a compound that blocks the activity of sugar phosphatase, an enzyme responsible for breaking down sugar phosphates. This inhibition can impact various biochemical pathways in the body because sugar phosphates are essential intermediates in several metabolic processes. Next is dextranase inhibitor. A dextranase inhibitor is a compound that blocks the activity of dextranase, an enzyme responsible for breaking down the polysaccharide dextran into its component monosaccharides, a process called dextranolysis. These inhibitors can be used to control this process potentially for various applications in the sugar industry and other fields. Next is antipruritic. An antipruritic is a medication or treatment that inhibits or relieves itching, which is often associated with conditions such as sunburns, allergic reactions, eczema, etc. The term pruritus is Latin for itching. These are the references. Moving on. Next is the bioactivities chosen for vanillin from Pass Online website. First is membrane integrity agonist. A membrane integrity agonist is a compound that promotes or enhances the integrity of cell membranes. Cell membranes, particularly the plasma membrane, are crucial for maintaining the homeostasis of cell, protecting them from the external environment and facilitating communication and transport between the inside and outside of the cell. Now let us know about homeostasis. So homeostasis is a self-regulating process that allows living system to maintain a steady internal physical and chemical environment even when the external condition changes. This is a figure showing the homeostasis process. These are the references. Next is anesthetic general. Now general anesthetics are drugs that act on nervous systems, causing it to be inhibited and resulting in the loss of sense of pain. They act on the central nervous system, causing reversible inhibition, resulting in loss of consciousness and sensation. Here, reversible inhibition is when an inhibitor temporarily suppresses an activity which can be restored once the inhibitor is removed. So, references. Next is carminatic. Carminatic bioactivity refers to a substance's ability to prevent gas formation in the gastrointestinal tract and ease the expulsion of gas, relieving gastrointestinal discomfort. These are the references. Next is phosphatase inhibitor. A phosphatase inhibitor is a compound that blocks the activity of phosphatase enzyme. These enzymes breaks down phosphoric acid compounds into a phosphate ion and an alcohol using water. Now, phosphatase also plays a key role in regulating signal transduction. Signal transduction, also known as cell signaling. When cells break down, they releases enzymes that can degrade proteins. Phosphatase inhibitor block these enzymes, preventing them from breaking down the proteins we are interested in and how they work. These are the references for phosphatase inhibitor. Moving on to our final bioactivity, that is respiratory analytic. Respiratory analytic is a medication that stimulates the central nervous system to enhance the activity of the muscles involved in breathing. Drugs like doxapram are primarily used to treat respiratory depression and respiratory failure. Further references for this slide. Finally, moving on to the conclusion of this project. Through computational chemistry tools, I explored the bioactivities of three key compounds, aspirin, menthol, and vanillin. Our investigations revealed a spectrum of potential effects contributing to a deeper understanding of these compounds and their implications across diverse fields. Now, the references. Though the references are mentioned in each page, but these sites deserve some special mentions. So, this is all about this project. Thank you everyone for lending your kind attention throughout my presentation. Thank you so much.